we can go.
So if everyone puts themselves on mute, welcome, massive welcome to Coffee Buddies at the Biotech Digital. My goodness, this is number 12, 13, 14, 15, Graham.
Yes, 15, well done.
15th event of this week. And my goodness, have we learned a lot? Yes.
Yes.
My goodness, we've met all sorts of people. We've explored the biotech entrepreneur's mind. We've explored how investors think, and we've explored networking. What's the art of networking? And we've done the XY factor, where we've had 12 companies present to MSD, and we've got the final part of that this afternoon at 6 o'clock, when we will announce the winners, and the winners will get a chance to go into our best and best of best round, which will be in December. So we'll be running a number of other rounds on the way to December, and then we'll pick the best of best. They'll come together, and then there'll be a trophy. We haven't quite worked out what the trophy is yet, but it's a bit probably like the glitter ball, but much smaller.
I quite like the Pointless Trophy, you know, on BBC, something like that.
Something like that, but we'll design that. And we've had fascinating characters come to talk to us. And remember, Coffee Buddies is all about entertaining, engaging and informing. And we've had a tremendous amount of compliments. So we're really delighted about that. And today is no exception. We have a fascinating person coming to talk to us. And I'd like to introduce you all to Fredrik. And maybe Fredrik, you can say who you are, what you do and where you are, which with that lovely red building behind you.
Absolutely. Hi, everyone. My name is Fredrik Rahm. I'm actually in my kitchen in Sweden, but the very lovely picture behind it is actually from our office at Pelago Bioscience, where I work, also in Stockholm. Our office is actually where we used to have our conference room, with the whole wall is this very, very nice painting from the Swedish archipelago. So it's a picture from that, and I really like this part of the painting with the little red cottage, because I think it gets a Swedish atmosphere. It's very Swedish.
I love it. Very fantastic.
Yeah.
Maybe you tell us about what your background is, Fredrik.
Absolutely.
How have you got to wherever you are, and you can tell us wherever you are next. But
absolutely. So I'm a organic chemist by training, and then I worked as a medicinal chemist in the pharma industry for many years, both in big pharma and in a small biotech setting. Always working with small molecules in lead generation. And I would say the reason to kind of start telling you how I ended up where I am right now is, I think I've always had an interest in how to understand how, in my case, then small molecules interacts with proteins. So to really understand how these interactions work and what then happens to the proteins, and how to kind of use that to develop drugs. That's been a real interest of mine. I worked a lot with structure-based drug design and these things. And then for many reasons, I moved around a little bit, and now I'm then working with Pelago Bioscience, where we take this to the next level. So instead of looking at how molecules interact with recombinant proteins, we want to really understand how native proteins, or how molecules interact with native proteins. So really understanding the proteins and how they work, and how to engage with them, and how to kind of modify them. So now I'm at Pelago Bioscience. We work with target engagement, and I work as a project advisor. So really working close with our clients to work with this fascinating field.
Yeah, maybe you could tell us a little bit more about what this what is target engagement, and how do you connect? How do you measure it?
Yeah, so target engagement. Of course, you can look at target engagement in different ways. The way that we look at target engagement is first you have the target, and what is the target? In our vocabulary, a target is a protein, that has some kind of function related to a disease. So again, as I've always in my career, I've looked at how to modify proteins to kind of get a disease modified. So target engagement for us is target engagement in a physiologically relevant setting. So instead of using recombinant proteins, we want to be able to look at these interactions in live cells, for example, or in tissue at the site where it really matters, where you kind of the end game, where do you want your molecule to act to really be able to help the patient.
And is there any difference, Fredrik, between engagement of a target in a pathological tissue than a normal tissue? So if I take a piece of synovium that is inflamed rather than a piece of ordinary synovium, is there any difference in target?
I mean, that could be the decisive. That could be a difference that is really makes the difference between a relevant target engagement for disease or a target engagement that isn't really disease modifying, for example. So we always, I mean, the technique that we work with, we are able to assess this target engagement in, we say, physiological relevance setting, but it's also a biologically relevant setting. So if you have if the biology that you're trying to modify is only present in certain tissues or modified in cells that are subjected to something, then you should look at that in that biologically relevant setting.
And have you ever looked, I'm just interested, have you ever looked at a time course of a model of, I don't know, animal model of disease and asked the question, what's the relevance between the plasma concentration and the level of target engagement?
Well, we haven't really done that in the way that we look at target engagement, because again, the technology that we use is to see that the compound actually engages the target. So it's not really, it's not a functional assay. It's built on that the interaction is there and something happens to the target. Right. But then, of course, since we can do this in a biologically relevant setting, the biology is still acting. So the time course could be that we could actually look at what happens to target proteins in a tissue or in a cell, for example, after the compound has been there. So kind of pathways being activated. Yeah. And you could see what happens to proteins in that pathway. Yeah. So in that respect, we can do time, you could definitely look at a time resolved experiment, seeing how the biology kicks in, and then you can see proteins being affected by the biology that you're affecting. Right. That's really something that's interesting.
And I mean, if you look at the life cycle of a drug, the 7 years it takes from zero to the market, what points can your technology be useful?
I would say the whole value chain, but we're looking at it traditionally. I think we started in I would say in lead optimization, where you might want to really confirm target engagement in a specific cell line or in tissue or from an in vivo study for a few compounds to kind of get confidence in really taking a few compounds further on. But our technology, the CETSA technology as it's called, has really evolved. So now we can use this technology for screening, so really early. Can use it for target convolution after a phenotypic screen, so also really early. But also get more and more projects that are towards preclinical, or we're also looking into clinical applications, as you briefly mentioned on tissue. So it's the whole drug discovery value chain, I would say.
Yeah, and I mean, if you go back to your time when you were interested in the structural biology, I mean, how is this working with modern structural biology thinking?
Well, I would say that this, thinking back to my career as a medicinal chemist driving projects before the CETSA technology was invented, it would have been really nice to again build confidence in what the compounds that you're designing and the biology that you're looking at, that these are actually matched, that your compounds actually engage your target. I mean, I've been in projects where we've done fantastic work in medicinal chemistry and great molecules, but then we just saw that, well, we don't engage the target in a relevant model. And of course, that is something that is broadly known in the pharma industry of lack of efficacy. That are you sure that your compound actually engages the target in the right, where it should be?
Yeah, I mean, there's always an interesting paradox between the way a chemist looks at the science and biology.
Yeah,
the chemist nearly always blames the biology as not being appropriate for binding to the target, and the biologist always thinks it's the other way around. I mean, it's a good way of untangling mechanisms of action.
Absolutely.
So tell us more about how that works.
Well, I mean, you can do this in different ways. I think basically you could go in either of two tracks, so to speak, so that you know the target that you're after. You have a disease. You have a theory on how to modify this disease. There's a target that you want to inhibit, and then of course you have. You develop your compounds and you optimize them for many things, but to make sure then that these compounds actually engage the target and couple this target engagement validation or this data to your functional readouts, that can really build confidence in that that the functional readouts that you get are really related to your compounds engaging the target. And it could be the other way around as well. If you want to, there's something going on and you don't really know, it's not, you can't see the trend with your targets. You can do, you can look on the whole proteome using the CETSA technology. What else is your compound doing? So to really get assessing 6,000 or 7,000 proteins in one experiment to see what your compound is doing apart from hitting your target of interest.
Yeah, which is fantastic. I mean, so if you were to, what questions does your technology answer? Then it answers the question, is there a biophysical binding? What else does it answer?
Well, it answers. I mean, it's a lot of things built into this data that you get. Again, the choice of what you started talking about, Tony, that the choice of matrix that you choose for your experiment, that it is a relevant setting. Is it in it's a cell line, or if it's a tissue? It also built in that the compound really reaches that. So, for example, if you have an in vivo study with dosed animals, and you can take tissue from those and then run the CETSA technology on that to see that the compound, you have target engagement in that tissue. That tells you a lot of things also from about that study, that your compound reaches the target in that in vivo study. So it is a I mean, there's a lot of cell permeability and everything like that, distribution, depending on how the experiment has been set up. So it is a full-featured experiment, so to speak.
That's fantastic. And what's the last question? What's the sort of type of customer you have? Maybe you can describe a customer and what they're looking for.
There's so many different customers, I would say. We have anything from major big pharmas wanting to run big screening campaigns with hundreds and thousands of compounds for a new project. It could be a small startup with having one compound with fantastically interesting phenotypic readout, and they want to see which targets this compound hits to start the company around this biology. Academic groups, you name it. That's one of the things that make my job so interesting and so enjoyable, talking to scientists and drug hunters in so many different settings.
Fantastic. Well, I mean, I know it's a groundbreaking and world fantastic technology, which could really give you insights that nothing else can. So fantastic. What I'm going to do is I'm going to ask Graham to curate some questions. I see some questions coming up. I think there's a fellow medicinal chemist called Gordon who has a question. So that's Graham, right. After I've found Gordon, maybe you could curate.
Yes, no, Gordon, if you'd like to ask a question.
Yes, so you've obviously done this many times now, and the first part of my question was how are you finding inhibitors versus agonists, and to the second point, what about other modalities? So what happens if you put Protac in that has a dual end, one that's going to stick to one thing and one sticks to your target? What does that actually look like? And have you done anything as detailed of that and found any surprises?
Absolutely, that's a very good question. I'll start with the first one. Just very briefly, then, what the method is all about. It's a thermal shift assay, so in cells, cellular thermal shift assay. So what we measure is a change in thermal stability on a protein target when it interacts with a molecule, or if it's in a live cell, of course, it can also be pathway-related effects. So you incubate your cells, for example, with a compound, and then proteins that directly interact with a compound, their thermal shift, their thermal stability changes, or something happens to phosphorylations or something, so that pathway, so downstream proteins are changed. And the thing is that we see both stabilizing events. So if a protein is engaged by the molecule, it could either be stabilizing or it can be destabilizing. And it's really, we have some cases where we and this is very target specific. So for a specific target protein, it could be, we don't really have that statistics yet. It could be that for that particular modality, agonists stabilize and antagonists destabilize. But we have it's a very complex. I mean, thinking of the protein in the cell with all the, I mean, it's full length, it's all the native and endogenous cofactors and friends are there. So if you will get a stabilizing effect or destabilizing effect is very specific to that cell line and to that protein. So what we typically see, for example, is for kinases, very often ATP competitive binders could have a destabilizing effect because ATP really fits in there. It's hard. I wouldn't say that you could use CETSA to say that a stabilizing effect shows agonism and a destabilizing effect shows antagonism. It's so both events would show target engagement. That's basically how we look at it. And then we come to degraders or Protacs. I mean, protein it's a fascinating modality, and we have done studies on this. And I think this is an area where actually the CETSA method shines, because depending on how you set up the experiment, you can look at both binding to the target of interest, so the target that you want to degrade, and of course the E3 ligase. But you could also look at degradation. And this is something that for example, in the mass spec in the mass spec version, you can really dig deep into what is a degradation effect and what is a binding effect. So the CETSA method can really get powerful data on degraders and Protacs.
Great. Okay, we've got another question from Katja.
Hi, thank you so much, Fredrik. It's a fascinating technology, very interesting. I wonder if you could clarify a little bit how you actually do the thermal assay. So you're looking at multiple targets. Do you express different proteins in a number of different cell lines and then look at transiently or permanently transfected cell, and how do you do that? And then I have a follow-up question.
Yeah, absolutely. The very short answer to, or the crash course in the CETSA method, we have two parts. So either it's the targeted CETSA, where you know you have a target of interest. So you do it in, well, basically we can, that's one of the power CETSA, that you do it in normal cells, or in the cells that are relevant for your biology. So normal cells is fine, as long as it's compatible with the disease or the biology you want to look at. So in the targeted format, we use antibody detection. So you would only detect the protein that you want to look at. And then we have another format, which is protein-wide, and then we use mass spec to detect the proteins. And then we can detect up to 6,000 or 7,000 proteins in the cell. So in both of these formats, it's actually very simple in theory. You take your you incubate your cells with or without compound, and then you heat shock the cells. And the heat shock then, when you heat, proteins, they denature. And the theory is that if you have a compound binding to the protein, you will either get a stabilizing effect. That's usually what we get, that a protein is more thermally stable when a compound is bound, a ligand is bound to it. So the heat shock will, less protein will be denatured on that heat shock. And then we measure how much, after the heat shock, how much of the protein is still soluble or still in its native form. And that's, by mass spec or by Western blot, or we also do use alpha LISA technology. So in the targeted formats, we do antibody detection. And it's basically quantifying how much protein is left in its native form or soluble form after heat shock. And then you use that data to process into target engagement. And then for the mass spec, it's protein wide, so we can do this. It's unbiased, so you can see effects on. I mean, you usually detect 6,000-7,000 proteins, but usually a compound interacts with, and maybe depending on your compound, of course, a handful up to 100 targets, depending on how selective your compound is.
And have you done cascades looking at effects, downstream effects for different targets in terms of agonists and inhibitors?
Yeah, we in the intact cell environment, so let's center this around the mass spec technology, where we look at many proteins. In the intact cell environment, we see proteins that are shifted both due to direct binding, but also pathway effects. And we have bioinformaticians in the company that look at the pathways. So we can get a lot of information out on the kind of the CETSA profile that you get, the binding or the engagement profile of that compound in a cell line. And that's very information rich on which pathways are being affected. So you could definitely use, I mean, we talk about target engagement, but it's of course also pathway engagement of your compound.
Very interesting. Do you have any publications?
Yes, absolutely. There were several publications. For example, using CETSA, the protein-wide CETSA for PROTACs, for example, and degraders. I can share some reference with you, definitely. And we have a brand new homepage. I'm really urging everyone to go to the Pelago Bioscience new homepage, which is very nice, I would say. And there's also a lot of information there. But let's connect after the meeting.
Great. I'd be interested in discussing some of our.
Absolutely. Great.
Fantastic. Graham, how are we doing for time? You're on mute, my friend.
It's the mix-up time. So yes, we're right on the stage of putting you into breakout rooms.
Before we do that, I want to thank Fredrik in case we lose any more. Fantastic. CETSA's the business. Please contact Fredrik.
Absolutely.
Okay.
Thank you very much.
Yeah, thanks very much. Okay, here we go. We're going to open those rooms up now. If you